Hi, everyone, and welcome to today's OTS webinar. Today, we're featuring Dr. Jinkuk Kim from the Korea Advanced Institute of Science and Technology, and is, in fact, like many of you, I assume, awake in the middle of the night. So we really appreciate that, Jinkuk, and we're really looking forward to your talk. So you can go ahead and take it away.
Okay, thank you, Erin and Ronald, for the kind introduction and also organizing this webinar. So my name is Jinkuk Kim at KAIST. And for those of you who haven't yet heard about our institute, KAIST, let me give you a really brief introduction. So KAIST is about 50 years old. It's not too old, but it's a science and engineering oriented university in South Korea, and it's in a city called Daejeon, which is about 2 hours by car from Seoul or 1 hour by train. And I have a long-standing history and have emotional attachment with KAIST because I spent 4.5 years as an undergrad. And after 16 years wandering around, I came back to KAIST about 4.5 years ago as a faculty member. And then what happened during this 16 years? So I was extremely fortunate to be able to learn from so many great mentors like David Bartel and Do-hyun Nam and Brandon Fry, Peter Park, and Tim Yu. I feel really I'd like to thank for their mentorship that made me who I am and where I am as a researcher today. So to give you a little bit more tour of KAIST, it's our central library, and there's open deck in the second floor, and where there's a nice cafe, and you can overlook the small pond, which is called duck pond, because there are ducks who think they are human. So I hope to see you at KAIST someday. So going into the main part. So I think many of you are have at least heard of Millicent study. And I'm not going to go into too much detail, except saying that I feel extremely fortunate to be able to work on this study with Tim Yu while I was a postdoc at Harvard Medical School with Tim Yu as well as Peter Park. And in this study, in a nutshell, so for a 7-year-old child with severe neurological symptoms, we were able to develop and validate an ASO therapy that was named Millicent after the patient name, and which we showed is able to correct or rescue the mis-splicing effect of a patient-specific mutation that was causing the really fatal condition called Batten disease. And we were able to go from the initial patient contact to the first patient dosing within a matter of a year. And this study was published already about 5 years ago. And as a follow-up study, we published another study just about a year ago. And this second study has really 2 points. So first point is that we developed a second N-of-1 ASO therapy that we named atipaxin. And the second point is that we developed a systematic, we developed a framework, as the title of the paper says, that can systematically identify patients for N-of-1 splice switching ASO therapy. So I was extremely privileged and honored that this paper was selected as a Paper of the Year award. And I know that this award is given not to me, but to all the authors of the paper. So on behalf of all our authors, we are truly grateful. And I have to say that I had a heck of a good time in Barcelona last year. And I think all of you did. And I took my entire lab there. And I'm sure they also had a great time. And it was just an amazing time. So I'd like to thank OTS again for for this award, also this opportunity to present today. So going into the main part, really the main part, in the Milasen study, it was, like I said, it's a really blessing for me to be able to just participate in this study. But thinking back, there's one unfortunate aspect of the study is the timing of the treatment. So as you might have heard, the diagnosis for the patient was delayed. And by the time we developed Milasen and start treating, the disease of the patient had progressed so far that the Milasen treatment was able to reduce seizure, which is the symptom of the disease. But we have to admit that we fell short of really changing the long-term clinical course of the patient, because as you might have heard, the patient eventually passed away. So when in the second time around, when we were looking for another second patient to apply this approach, we intentionally looked for a patient who were early stage in their disease progression, or ideally in the pre-symptomatic stage. So to make the long story short, with the help of Brad Margus, who is the founder of AT Children's Project, which is the foundation for a disease called ataxia-telangiectasia, or AT, we were able to find such patient, 2-year-old child with this disease, so AT, to talk a little bit about this disease. AT is in many ways actually similar to Batten disease. It is a neurological condition, severe neurological condition affecting mostly the cerebellum, which cause ataxia, and it has some eye symptoms, which is a telangiectasia, but it's very mild. So it's mostly the cerebellum degeneration, and it's fatal, and it's orphan condition without any available therapy. And genetically, it's caused by allelic loss of function of a DNA repair gene called ATM. So it's an autosomal recessive condition. The severe symptoms of this disease, which are mostly neurological, is known to begin at around age 5. But this patient was unique. It's different from other patients in that she was accidentally actually diagnosed by a newborn screening. So how it happened is that, so she was genetically confirmed diagnosis well before age 5, right after birth. So how it happened is that AT patients is known to have mild immunodeficiency, which is almost asymptomatic immunodeficiency. And she somehow got into one of the immunodeficiency newborn screening program and found to have a mild immune deficiency. And to follow up on that immunodeficiency, whole exome sequencing was performed, which showed she has AT. She is, as you can see here, she is found to have she is found to be heterozygous, compound heterozygous for ATM known loss of function mutations, both of which are null mutation, which is known to be completely loss of function. So based on this, we could expect that she is a classical severe AT patient as opposed to the mild AT patient who have some hypomorphic mild mutation, but she had both of her mutations were complete loss of function mutation. So the natural history of the classical AT, as you can see here, is that they are neurologically almost asymptomatic before age 5, but there is a steep decline, neurological decline from age 5 to 10. And they are, from around age 10, they are unable to walk and they have to use wheelchair. And most of the patients, unfortunately, pass away around this age. And since there is no available FDA-approved treatment, as you can imagine, the family of our patient was really frustrated because knowing this natural history of the disease, and there's nothing they can do. But when we looked at their mutations, it was found that one of her mutation, which is the maternal mutation, was amenable to rescue by the Millicent approach. So we call it ASO amenable mutation variants. So looking at this variant, whereas our patent patient mutation was insertion of a long DNA element, retrotransposon, in the middle of an intron of a critical lysosomal function gene, which leads to mis-splicing, which was able to be suppressed by the treatment of Millicent. This child's mutation was a bit different in that it's not intronic insertion, it's an exonic mutation, and it's not insertion, it's a single nucleotide variant. So looking a little bit in detail, this C to T mutation in the middle of an exon was also can be considered as a missense mutation because it has a amino acid changing effect, but this coding impact is found to be benign. But it has a strong gain of splicing effect because the C to T mutation creates this critical GT dinucleotide motif that is necessary for the splice donor site, as many of you already know. And this was leading to mis-splicing in a way that it excludes 64 nucleotide of this critical exon. And because the 64 nucleotide is not multiple of 3, it leads to frame shift and premature translation, and which triggers nonsense mediated decay and loss of function of this maternal allele. So, but interestingly, this mutation was already experimentally found to be amenable to ASO by this paper published a long time ago in 2007. So what they found is that for a patient who was homozygous, so our patient was compound het for this mutation, but this patient was homozygous for this same mutation that our patient had, and they showed that it caused mis-splicing, and they was able to design a morpholino-based ASO. And when they treated this morpholino-based ASO against the patient fibroblasts, they were able to see that it can suppress the normal splicing and rescue some of the normal splicing. And it can rescue on a dose-dependent manner the protein expression as well. However, this is very nice. However, as you know, morpholino-based ASO is not clinically validated for CNS diseases. It has been used for muscle diseases, DMD, but for CNS diseases, it has never been used, to my knowledge, clinically. Presumably because morpholino has low solubility, which is not suitable, that makes it unsuitable for intrathecal administration. So we redesigned this set of ASOs based on the same chemistry as the one used for nusinersen and milesen, the full two methoxyethyl and phosphorothioate backbone, and conducted the screening based on RT-PCR. So Diana and April are really amazing people and good friends of mine. And when they screened using in two different rounds of the screening. And to make the long story short, we tested many ASOs and we were able to identify the lead candidate AT008, which later also passed all the validation, including animal toxicology, and became the final lead candidate, which we renamed to atipexin. And to talk a little bit about more about this lead candidate, atipexin, as you can see here, directly suppress this mis-splicing by complementarily binding to the against the novel splice site. And when we tested on the patient fibroblast, as you can see, it can rescue the it can suppress the abnormal splicing and restore the normal splicing very effectively in a dose-dependent manner. This is a little bit of oversimplification because we used actually allele-specific RT-PCR assay, where we used allele-specific primer, which is a little bit complicated. I'm not going to go into the detail, but this way we could specifically amplify the target allele when our patient cell is actually compound het for this mutation. Okay. So it specifically amplify only the target allele. And Tojo is an old colleague of mine back in the U lab, who is a really rigorous scientist who used to be a postdoc, but now runs his own lab in Japan. And what he found is that atipexin rescues not just the mis-splicing at the RNA level, but also rescue the function of the ATM itself. So ATM is, like I said earlier briefly, it's a DNA repair protein. And what it does is that when there is a double-strand DNA break from external stimuli like ionizing radiation, it detects this break and induce the repair downstream pathway. And the key effectors of this downstream signaling include p53 and COP1, which gets phosphorylated by the ATM. And when Tojo Tojo irradiated the parental fibroblast, which is obviously phenotypically should be normal. The downstream effectors the signaling gets activated, but when he irradiated the patient fibroblast, who is completely lacking ATM function, as expected, there is no activation of downstream signaling. But when we treated atipexin, we were able to see a rescue of the downstream signaling. However, as you can see, the extent of rescue falls a little bit short of the control. Okay, as you can see, the signal is a little bit weaker with the patient fibroblast treated with ataxia. But when we compared the extent of rescue, not only to the parental control, but also to the mild AT patient, we were able to see that the ataxia treated treatment rescue the downstream signal beyond that is seen in mild AT patient. And because these AT patients are substantially mild, many of these patients live into their 60s. So we thought that this ataxia treatment can have a meaningful clinical impact. So with this validation data for ataxia, and with the financial support from the AT Children's Project, we were able to proceed to the IND enabling studies and N-of-1 clinical study. And I'm showing it as a single slide, but as you can imagine, if you have done this process, it's just an unimaginably huge amount of work. And it took really, it wouldn't have been possible without the really dedicated people like Aubrey, who is a good friend of mine, and is a really amazing bioinformatician and also project manager. And Claudio, who is also is a really brilliant physician scientist, and so many other people, and of course Tim. Because of these people, this was possible. So we gave the first dose to the patient in January 2020, which is about already 4.5 years ago. And we have been, the patient has been treated roughly every 3 months. And up to now, fortunately, without any severe drug-related adverse event. So I'm just going to show you some articles. So this is 1 month after the initial dose. And this was about 1.5 years after the initial dose. And this is 3.5 years after the initial dose. When the paper came out, this article also came out. And now it's already a year ago, so now she should be about 7-year-old. But I personally have no information about the most updated status of the patient. But at least until 6-year-old, she hasn't experienced the steep neurological decline that most other AT patients experience between age 5 to 10. And so we need longer, it's encouraging, but we need, of course, longer term treatment and follow-up to know for sure whether ataxia had any benefit. And we are at this point hoping that this newborn screening and pre-symptomatic genetic diagnosis and treatment can really change the long-term clinical course of this patient. So based on our milestone and ataxia study, at FDA published a few guidance on N-of-1 ASO therapy that is applicable for fatal, rare, and orphan conditions. And after this, our papers got published, and after this guidance got published, we've been getting a lot of emails from families and clinicians asking whether their patient can also try this approach. But one limitation that I have to note is, one important limitation is that this approach is not, of course, not for all patients. Because as you can see, it can be applied to only a certain subset of patients with a very specific type of mis-splicing mutation, like the mutation in our found in our patent patient or in our AT patient. To provide a quick feedback to those emails inquiring about the ASO amenability, and also in order to be able to perform genome-based screening of patients who can be candidates for this therapy, and also in order to estimate the fraction of the patient who can be candidates for this therapy, we felt that we really need a framework for systematically identifying patients with ASO amenable variants. So to do that, we expanded our collaboration with the AT Children's Project, which also funded our AT study. And we analyzed the whole genome sequencing data, their whole genome sequencing data of 235 patients in their patient registry who were clinically diagnosed with AT. And so these patients were clinically diagnosed, but they were a large fraction of these patients were actually lacking the annotated genetic full diagnosis. But by doing this comprehensive genome analysis on the whole genome sequencing data, including the small SNVs, single nucleotide variants, and copy number and structural events like transposons, we were able to establish the full genetic diagnosis on almost all patients. So we were by identifying the biallelic pathogenic, high confidence pathogenic mutations that is required for the establishing the full genetic diagnosis for autosomal recessive conditions like AT. And around this time, I moved from the U lab to KAIST to set up my own lab. And most of the genome analysis that I'm going to talk about from now on was done by my student, CJ Wu, who recently graduated, and who is a very talented MD PhD student. So to identify the ASO amenable variants from the pathogenic mutations found in the AT cohort, we developed a mutation classification system that we called taxonomy of ASO amenability. So what it does is it's really similar to, in a way, ACMG classification that is used for pathogenicity classification, but except that it is for not pathogenicity, but for ASO amenability. So given a high confidence pathogenic variant, it can classify whether it is ASO amenable or not. But if it is classified as ASO amenable, it can tell whether it is high probability one or a medium probability one. So it's a 3-tier classification system. So I'm not going to explain too much detail because it's can get very technical, except saying the two main ideas underlying this classification system. So the first idea or first criteria is sort of a negative selection. So if a variant destroy either the coding function or the canonical splice site motif, then it is, no matter what, it's unlikely to be amenable. Because, for example, large deletion of a gene cannot be rescued by ASO, or frame shift mutation, or nonsense stop gain mutation, or severely damaging missense mutation cannot be rescued by splice-switching ASO. And if the mutation is in the GTAG, the splice site core dinucleotide motif, then the canonical splice site gets completely destroyed, then it cannot be rescued. But if it does not damage either the coding function or canonical splicing, then it can be probably amenable. If you think about our pattern disease patient mutation or AT patient mutation, they do not disrupt the coding function or the canonical splicing because they are either in the middle of an intron or they are benign missense mutation in the middle of an exon. And anything in the middle is classified into assigned to the middle tier. And the second idea or second criteria can be considered as a positive selection criteria. So if a mutation, let's say, does not induce any mis-splicing, it cannot be rescued by splice-switching ASO, right? If it doesn't have any splicing impact to correct, right? But if the mutation has any mis-splicing event, gain of splicing event is easier to be rescued by ASO than exon skipping event or intron retention events, because you can simply suppress, as we did for with that Pexen, suppress the novel splice site for a gain of splicing. But for exon skipping or retention, you have to strengthen a weakened splice site, which tends to be more technically difficult. So if you can imagine that for example, the Batten disease patient's mutation or the AT patient mutation, they satisfy both of these criteria. They have no impact, no damage to this function, and it's a gain of splicing. So it's classified as a probably amenable mutation. So let me get back to this a little bit more in detail later. But if you think about it, this classification is a little bit it's a little bit subjective classification because it's based on our concepts. And we use some predictive models like splicing prediction based on Splice AI and MaxSenseScan and some Revel for coding impact prediction. And we use somewhat subjective cutoffs. So we thought that it is necessary to experimentally validate this taxonomy. And it is inherently very challenging to do this validation because in order to prove that a given variant is ASO amenable, you have to really develop ASO and ensure that it works. So it's a lot of work. And it takes a lot of dedication. But what and Bo-Soon is an old colleague of mine back in the U lab, and is extremely talented postdoc. And what he did is that there were 17 unique, probably ASO amenable mutation in the AT cohort. And out of the 17, for 11 probably amenable variants, he was able to establish mini-gene mis-based mis-splicing assay and was able to confirm that all 11 of the 11 that he tested, he was able to develop effective ASO that can rescue the mis-splicing. So based on this experiment, we were able to say that the probably amenable mutations that is predicted by our taxonomy is likely to be actually amenable to ASO experimentally. So when we applied this taxonomy to the mutations found in the AT cohort, we found that about 9% of the individuals have at least one of the two hits in those patients is a probably ASO amenable mutation. And these mutations can be subdivided into 3 different types. So first type is mutation that is very similar to our patented patient mutation, in that it is a long intronic insertion, which leads to, as you can see, it's a gain of splicing. So it satisfies both of the criteria for probably amenable mutation. And the second type is actually very similar to our AT patient mutation, that it is either synonymous or benign missense mutation, so it doesn't disrupt the coding function or the canonical splice site. At the same time, it has a gain of splicing. And the third type of the mutation is actually a new type, which is a deep intronic mutation, but it's a single nucleotide variant that leads to gain of splicing, and which result in the activation of inclusion of a poison exon in the middle of an intron. And it turns out to be the most frequent type of probably amenable mutation compared to other types. But maybe it is because there is a founder mutation in this cohort, where this particular mutation was found as a heterozygote in 13 patients out of the 235 AT patients in this cohort, mostly found in the UK heritage. And as you can see, this is a single nucleotide variant, deep intronic variant. It's more than 1,000 base away from the nearest exon. And this A to G mutation somehow activate this spliced donor site, cryptic donor site, and indirectly also activate this cryptic acceptor site in the middle of this intron, which leads to, as you can see from this RNA-seq data, for a patient who is heterozygous for this mutation, compound head for this mutation, you can see the inclusion of this poison exon, whereas in a patient without this mutation, you don't see the inclusion of this poison exon. So we developed this ASO that can effectively rescue this mis-splicing, but we didn't choose this ASO as a candidate for clinical development, even though if it is developed, it can benefit more patients, because this turns out to be a mild mutation, because this mutation, this inclusion, this mis-splicing is leaky. This mutation has some residual, leaves some residual function that many of the patients who harbor this mutation, they live into their 60s. So there's less critical need or urgency compared to our AT patient. So so the first major conclusion from this AT whole genome study is that at least for the AT cohort, about 9% of the patients have what we call probably amenable mutation. And if we include the possibly amenable mutation, and it becomes 15%. So I haven't explained a little too much about this possibly amenable mutation. So I'm gonna show you a few examples of possibly amenable mutations. So first example is a synonymous or benign missense mutation in the middle of an exon. So this mutation, by first criteria, it doesn't damage the coding function or canonical splice site motif, but it does not induce gain of splicing, but it induces other types of mis-splicing. So it's considered possibly amenable. But if it induced gain of function, it would have, of course, classified to the probably amenable mutation, just like the ATPXN target mutation. The second example is a missense mutation in the middle of an exon, but it's incompletely damaging the coding function. So in this case, it is damaging to some extent the coding function. So it is classified into this middle tier, right? And then no matter what mis-splicing event it causes can be gain of splicing or exon skipping or whatever, it's possibly amenable because it has some damage. Even if you rescue, you are not 100% sure that this is going to be a functional protein because of the missense mutation. And the third example is a near intronic mutation that partially disrupt the canonical splicing. It can be near the branch point of acceptor, or it could be near the splice donor site or splice acceptor site or splice donor site. But then it partially disrupts the canonical splicing. So it's a moderate damage. Like the previous example, no matter what mis-splicing event it caused, it's a possibly amenable mutation because this is difficult to be rescued because the canonical splice site itself is damaged. So even if you use ASO, you might not be able to resuscitate that damaged splice site. So one thing I wanted to note here is that the possibly ASO amenable mutations it means that there is a theoretical possibility of rescue by ASO, but there is much less chance of successful ASO development. So I would not necessarily recommend following up on this possibly ASO amenable variant unless you have a very strong reason to follow these mutations. So again, the first conclusion from this study is that about 9 to 15% of the mutations of the patients in the at least for the AT cohort have the can be candidates for the Millesen approach. And we wondered whether this estimate can be generalized to other genetic diseases. And we did some meta-analysis on the ABCA4 deficiency, the sequencing studies. And we were able to conclude that a similar fraction of patients are amenable. But I have to note here is that this estimate of the prevalence of ASO amenable variant is bound to be different for different diseases and even for the same disease with different ethnicity because there are different ethnicity-specific founder mutations that could swing this estimate by a lot. So analysis of recently the big rare disease whole genome sequencing studies that became recently available, like the Genomics England study, will be able to give more accurate estimate on the prevalence of these amenable mutations in many different diseases, which I'm going to talk a little bit more about later. And the second major conclusion from this study is that because most of these, especially probably amenable mutations, are deep intronic mutations, either deep intronic insertion, deep intronic single nucleotide variant, which usually cannot be detected by whole exome sequencing. When we simulated how many probably amenable mutations we would have detected if we did whole exome sequencing instead of whole genome sequencing, we found that if we did whole exome sequencing, we would have found 5 times less probably amenable mutations. So, so we call it ASO therapeutic yield, the chance of detecting, finding the ASO amenable mutation. And it's instructive to compare this concept of ASO therapeutic yield to the concept of diagnostic yield, because when it comes to genetic diagnosis, whole genome sequencing does not add too much value on top of the whole exome sequencing because most of the pathogenic mutations are in exons. If you do just whole exome sequencing, you can identify at least one mutation from almost all patients, and you can find 2 hits. You can give full genetic diagnosis for about 90% of the patients. So by doing whole genome sequencing, you can find one additional mutation in about 10% more patients. So, these additional 10% of the mutations that you can only find by using whole genome sequencing tends to be deep intronic mutation that leads to gain of splicing, which is the typical probably ASO amenable mutations. So I'd say that whole genome sequencing may not add too much value to the genetic diagnosis, but it is critical for identifying patients for NF1 splice-switching ASO therapy. So to summarize a little bit, looking back and looking forward, our milescence study provided, provided proof of concept of this approach. And then our AT study really demonstrated the repeatability of that milescent approach and the applicability of this approach even for a pre-symptomatic patient for better long-term clinical outcome. And it also developed a framework for systematic patient discovery using AT as an example disease. The next challenge is going to be applying the framework that we've developed to thousands of other disease cohorts. And you can use the recently available rare disease genome studies like Genomics England, or there's a Korean version of Genomics England, which includes really tens of thousands of affected patients' whole genome sequencing data. But the limitation of this approach also is that most of this we will be able to find so many patients who have amenable mutation, but these patients are all most of them are post-symptomatic patients because they were included in this cohort because they have symptoms, most of them. So the final challenge, I believe, at least in terms of the patient identification, is to apply this framework for to the newborn genetic whole genome sequencing screening. So, but then yet another challenge, once we find enough of these patients with amenable mutations, how can we provide treatment for these patients? So, but thanks to the organizations like the NF1 Collaborative and Dutch Center for RNA Therapeutics, and also, of course, and Loren Foundation, who recently published their first successful case, first case. And more patients have access to this treatment. But given that the cost of this lifetime cost of NF1 therapy still is in the range of $1 to $2 million per patient, truly broad access to therapy is possible when the cost of the NF1 therapy goes below maybe, say, $1 million, which is the lifetime cost of symptom management. And it is possible through innovations like process optimization and regulatory reform, like the class approval of this ASO drugs, which I am very optimistic given all the effort that is being done by the foundations and the organization that I just mentioned. And at that point, when the costs get lower than the cost for symptom management, the cost can be covered, hopefully, by insurance, and it can be really applied to hundreds to thousands of patients. So this is my last slide. And going back to the genetic testing of our AT patient, for 90% of rare diseases, there is no approved therapy, so which was the case for our AT patient. But if we can add another column in this report, the ASO amenability classification. And we then, even though it is theoretical possibility because there is no capacity to pursue all the cases, we can at least increase the theoretical therapeutic actionability of this genetic reports and expand our capacity to treat all these patients. So with that, I'd like to thank all the people who've contributed to this work, most of whom I've mentioned along the way, but notably and most importantly, Tim Yu, who has been really the driving force and central to all this effort. And I personally thank him for all the mentorship. And I'd like to thank the families of our AT patient and the Batten patient, and the AT Children's Project, who funded most of the effort that I described today, and the members of my lab and the funding source. Thank you so much, and I'm happy to take any questions.
Yes, Jinkuk. Thank you. I think that was an excellent presentation. And I think, once again, a big applause for winning the Paper of the Year award in 2023. So for the people that were there and not there, congratulations. So if you have a question for Jinkuk, please use the Q&A function. I think I will start with a question. So, especially for the AT patients, you have a very small window of opportunity, because I think what you mentioned during your presentation is that the first symptoms or the first decrease starts already at the age of 5. And also what you mentioned before is that most people don't even know that they have a particular disease or a particular mutation. You only discover this when you experience the first symptoms. So are you so should we do more newborn screening, or should we increase the newborn screening, or should we do a whole genome screening for every newborn person in the world? And the second question is, how much time do you think that we need to develop an ASO? And do we have this time between the identification of the mutation and the first symptoms or the decrease in symptoms?
Okay, thank you. So if I understood correctly, so I think that timing of treatment is absolutely one of the most important factor in the success of the treatment. So that in that sense, picking a patient who is early in their disease progression is really of a paramount importance. And but at this point around, we don't have capacity to treat so many patients. So if we work with the foundation or large database like Genomics England, we can find a patient who happened to be diagnosed pre-symptomatically, like our AT patient. And if we treat those patients that have the most expectation, the highest chance of success, clinical success. But ultimately, like I mentioned, and also you pointed out, that the newborn screening is the holy grail, and but it's also very challenging, because then you are dealing with individuals without any clinical diagnosis, then you don't know where to look. You have to look at all 30,000 genes, and there are a lot of false positives. So, but once we have a better, more comprehensive clean bar, like the known pathogenic variant database, then we can reduce the false positives, because the source of false positives is in many cases is the prediction. So the coding impact prediction or splice prediction became very accurate, but they are not perfect. So it produces a lot of false positives. But the once we have much richer pathogenic mutation database, we can reduce the false positives. And we should, I think, start thinking about this newborn screening and looking for these amenable mutations going forward.
Yeah.
Yeah.
So I think already some other patients with the same mutation are also treated now, right, for AT? Can you comment on this?
Could you say that again?
I think the same mutation. So I think multiple other patients are also treated with atipexin right now.
Right.
Is it correct?
Yeah, right. So there is, I haven't mentioned, but there's another patient with the identical mutation found, and the patient is also being treated.
And can you mention how you determine the dosing regime? Because it must be quite difficult to treat a child of 3, 4, 5 years old. So how do you determine the dose interval and the dose frequency and the dosing?
Right. So there are, I think there are two standards. So I have to admit that it's relatively so the one way that we could estimate the target dose is based on the dose used for nusinersen, and nusinersen, as you know, it used to 12 milligram, and it is targeting the spinal cord, and we know that the distribution of ASO upon IT injection to the spinal cord or to the brain is not the same. So there are 1/3 of ASO distributed to the brain compared to the spinal cord. So we, based on the simple backup envelope calculation, we reasoned that we should go 3 times higher than nusinersen in order to achieve the same ASO concentration in the brain, which is the target tissue of our patient disease. So that's how we determine the target dose. But also another way is based on, of course, the toxicology study when we established the NOEL and LOEL, and we know we can do the conversion between rat and human, and we can establish the dose, the non-toxic dose and the toxic dose levels, and that's another guidance. And third way is that we did the dose escalation to the target dose level every other week, increasing the dose. Because it's a first in human study, we have to also prepare for the worst case, that is it's more toxic than previous drugs. So, but then once it is well tolerated, then and we have more safety margin based on the rat toxicology study, then we can increase the dose a little bit further. So that's why we went all the way up to 63 milligram, which is 50% higher than 42 milligram that we initially settled on, based on the 3x from the nusinersen, after accounting for the molecular weight difference. So so that's how we decided the dose.
Do you have any idea how the patient experiences all the intrathecal injections? Is it okay? Is it difficult to deal with?
Oh, so yeah, so intrathecal injection is a very high threshold because yeah, it's an invasive procedure, but for our patients, they because they are young, they had to go through the anesthesia to be able to safely inject the drug. So that's so because it's invasive procedure, so we think it is suitable only for severe life-threatening conditions. Right. Yeah.
Okay, I will move to some questions in the chat. So there are quite a few. So and it's also already past the hour. So maybe a few shorter answers. I think you already discussed the tox study in red. So did you? So is the splicing event corrected by antipexin conserved in animal species? So did you do any animal testing, not only for the tox but also for a molecular effect to test the molecular effect?
Right. Yeah, that's a good question. So for both milestone and antipexin study, of course, the mutation is not conserved between species. It's not even shared between human individuals because it's a patient-specific mutation, right? So only that patient, not other even human individuals, have the same mis-splicing event. So so we cannot, of course, see the efficacy in the animal studies. We can only see the adverse events and estimate adverse event from the rat studies. And that's so we have to rely on the cell-based assay to rescue the mis-splicing and the phenotype, like it showed the downstream signaling of ATM. But if there's enough time, but we are running out of time because of the patient disease progress fast. But if we have time, then we can engineer that mutation, patient-specific mutation, and the context of that mutation into the mouse, and can test also the efficacy in the mouse model, which was tried for some other cases where there was Niemann-Pick C twins who unfortunately passed away before we had a chance to treat, but their parents actually made a mouse model of their twins mutation. Yeah, so that's possible, but it takes time because yeah, it's not feasible.
I can imagine it might take you over a year to have a good mouse model.
Right.
To phenotype it still. So
right.
good. A few short answers. Great talk, very inspiring. For rare diseases, unlikely to be treated by ASOs, are there any new technologies, for example, CRISPR or base editing, to cure the disease? So how do you compare your ASO treatment versus CRISPR-Cas or via versus base editing?
Right. Yeah, that's a great question. So I think that yeah, different modalities have different advantages. So the biggest advantage, and so they are mostly constrained by the delivery method. So so for example, CRISPR, you have to deliver Cas protein, which is to do so, you have to use either AAV or mRNA, lipid nanoparticle, which can be delivered only to a certain tissues, like liver. Maybe you can use some maybe some other tissues, but by using AAV, but that entails other issues, like the manufacturing is very expensive and there's some safety issues. So so I think, you know, the gene editing is amazing. It can correct any mutation, almost all mutations, but you have to the delivery is the challenge. But the ASO, as you know, can be delivered as a naked form to the central nervous system, and it's working. It's validated by nusinersen. So so that's the biggest advantage of ASO, is that it is proven to be effectively delivered to the CNS without any expensive manufacturing like LNP or conjugation or anything. So that's why we can do N of 1 because the manufacturing cost is really low compared to others. Yeah, and sRNA is great, but it has been working only for liver with the GalNAc conjugation. But there are technologies being tried, like the lipophilic conjugation, or as you know, it's di-sRNA, but they need to be validated clinically before it is more widely used as an N-of-1 setting. Yeah, thank you for the question. It's a great question.
Jokesh, so congratulations again for the Best Paper Award. So my question is about the cost of the therapy, which you shared in the last slide. Do you have an estimate about what proportion of the cost is from the drug manufacturing?
Oh.
So how much is the drug development and how much is the manufacturing of the drug?
Right. So I think the most part of the cost, if it is $2 million, a lot of it is clinical cost of running the clinical trial and measuring the PK of the patient every time we inject of the drug in the CSF, and establishing and the PK assay, and another big chunk of the cost is the of course rat toxicology GLP study. So I think if the class approval of these ASO drugs is realized, then we don't have to go through the expensive rat-based toxicology study for every single one of these N-of-1 ASOs, which will reduce the cost by about $500,000 immediately. So that's I think the biggest advance in reducing the cost. But manufacturing it depends and the actual drug substance manufacturing, drug product manufacturing, and there are a lot of quality control. So the raw material cost is very low, and it is manufactured by the same instruments that is used to manufacture like the primers. And so it's if you go down to the very bare cost, it's low, but there are a lot of quality control, and you have to do stability testing, and then you have to release testing, and it costs a lot of money. But if you optimize that process for an N-of-1 setting in small batch manufacturing, then you can, I think, reduce the cost by much. But yeah, it depends on the manufacturing companies and so forth. It's hard to give a one number, but it's a relatively small portion compared to toxicology.
Yeah, a question from your colleague, Boksun. Great presentation. So you mentioned that the individuals enrolled in the Genomic England and the KBB studies are post-symptomatic, so implying that the optimal treatment window might have passed. Given this, what value lies in examining these cohorts, and what insights can we gain from these studies?
Oh, okay. So yeah, so it's so there in the Genomics England, there are rare disease cohort and then non-rare disease cohort and so forth. But looking at the rare disease cohort, they are mostly post-symptomatic patients, and so it's very difficult to find patients suitable for therapy in terms of the stage of the disease from that cohort. But just like our AT study, it will be for a generalization of our AT whole genome study, where we analyzed post-symptomatic patient whole genome sequencing so that we can systematically identify this potential ASO amenable mutations. And once we can characterize the prevalence of each mutations and what are the founder mutations and what are the relative prevalence of this amenable mutation in each disease cohort. And it'll be informative in prioritizing our effort to different diseases, because we want to go after a particular disease with a lot of amenable mutations, a lot of founder mutations, and we can use the same clinical protocol and the functional assay for that same indication. So it'd be helpful just for us to have an optimal strategy.
I'll do the last two questions because it's already 10 minutes past the hour. So Lakshmi, excellent talk. have the downstream DNA damage functions been assessed in post-treatment? So in the patient itself, because I think you saw a very nice rescue in your cell model.
Yeah, I'm sorry. Could you say it one more time? I couldn't miss it.
Have the downstream DNA damage functions been assessed in post-treatment?
Oh, I see. Post-treatment. So, so I'm not sure if I understood correctly, but so we used the fibroblast and what we looked for is the downstream signaling of ATM after inducing the double-strand DNA break. And there is a functional expression of ATM. There will be downstream phosphorylation of P53 and other effectors. But we didn't actually necessarily look at the damage site itself, if that is the question. I'm not sure.
Yeah, I think the question is, did you also assess this in the patient itself? So in blood or CSF, did you check the downstream targets?
Okay,
so
to
see whether there's a functional effect also in the patient.
I see. So that's a great question. But given that, so it requires for us to get the samples from the patient. So it'll be challenging to so that would be very useful to know that whether that there's an in vivo sort of a test of efficacy of our drug, but for both the Batten patient and AT patient, the target tissue is the brain, and we cannot sample the cells from either from brain or any part of the central nervous system, so that which makes it difficult for us to see the effect, measure the effect of the drug in vivo in the patient.
Very last question. So I think ATM is also involved in many cases of cancer. Do you think that an antisense therapy will also help to treat cancer?
Right, that's a good question. Yeah, so it's involved in some, to my knowledge, its susceptibility, even heterozygous loss of function of ATM is confers a susceptibility to some cancer, like blood lineage cancer or like some breast cancer or something like that. But it's really boils down to whether we can deliver ASOs to the target tissue. If it is breast cancer, it is not possible. If it is blood cancer, it's not possible to effectively deliver ASO given our clinically validated any of the platform. So as you know, ASO can be effectively delivered to only a set of tissues like the CNS, eye, or liver, or so if the cancer is expected in those tissues, it's possible. But if not, it's not, I think. Thank you.
Thank you very much. So I think I give the word to Erin.
Great. Well, thank you so much again, Jinkuk. That was an amazing talk. It's always inspiring to see what you guys are doing and what is coming of your research. And so congratulations again on your award. And we would just like to remind everyone who is still online or who is watching the broadcast that we have another webinar coming up on August 29th. And it's featuring Laura Croft and Mark Fisher, and is also a highlighted paper from our partner Turner Nucleic Acid Therapeutics. So please do sign on for that. And then we'll have another session on September 19th. And then we have a bit of a break because we have the conference coming up. And then we'll be back in the late fall. So thank you again, everyone, for tuning in. And thanks again, Jinkuk, for your amazing talk.
Thank you so much, everybody. Thank you, Erin, and thank you, Ronald.